Okay, so I want to thank the organizers for this opportunity. Actually, it forced me to think about replication in disease, which is very timely for me because I just moved from Florida State University, where I have been spending 50% of my time doing teaching and service to the university. And I've moved now to a soft money small institute in San Diego, where I want to use that 50% of my time to collaborate with people doing translational research. So it was a good opportunity to think about replication timing in disease. So what I've shown you here is a hypothesis that I've been working with actually since I was an undergraduate student. And basically, for the purposes of this meeting, I would guess that most of you, when you think about DNA replication in disease, you think of genome integrity, and enzymes that maintain genome integrity. And this audience probably thinks of DNA methylation, DNMT1, and the pathways that maintain DNA methylation during replication. But my point here is that it's not just DNA that replicates, it's the entire structure of the chromosome that duplicates. And we now know that different types of chromatin are assembled at different times during S phase. So my argument is that it would seem kind of crazy to think that Mother Nature would squander this incredible opportunity to either maintain or alter epigenetic states. The problem, of course, has been that we haven't understood how replication is regulated, so we haven't been able to manipulate it to test this hypothesis. And today I'm going to show you that those obstacles are starting to break down. Okay, so fast forward a few decades, and we now have methods to measure replication timing genome-wide. We have high-resolution methods and single-cell methods. Here I've shown you our simplest method, where we collect the DNA that's replicated either in the first half or the second half of S phase, and then display a log-2 ratio of early to late. And you can see that these are raw reads in 5 kb bins, and you can see that replication it does proceed in these synchronous waves of large segments of the genome that replicate either early, middle replicating, or late replicating. And we can then, sorry, I had to get the forward button here. We can then divide the genome into different into segments or domains, and ask questions about the sizes and numbers of the domains, etc. And you can see that there are these regions that replicate relatively synchronously, and they're punctuated by what we call temporal transition regions, where we believe for the most part replication is proceeding unidirectionally at 2 kb per minute. So to summarize many years of work here, what we found was that this program is incredibly robust to all sorts of insults, downregulation of chromatin regulators, etc. But as soon as we differentiate stem cells into different cell types, there is a massive reorganization of replication timing. And overall, in all the cell types we've looked at, about half of the genome changes during differentiation or during disease. And these changes occur in units of 400 to 800 kb. So just as an example, if you look at this large early replicating domain, parts of this will be found to replicate late in other cell types and switch back to early. And always these units have the same boundaries in different cell types when they switch, and they're always in this range of 400 to 800 kb. So this was something we discovered early on in 2008, our first genome-wide publication. And this gave us the first indication of what might be a unit of regulation of replication timing, which we call replication domains. And most of you know these changes are coordinated with changes in transcription, chromatin structure, 3D organization. And something that's not so well publicized is that we showed that when you make iPS cells, the genes that are difficult to reactivate in iPS cells are located within late replicating regions that also are difficult to reprogram back to early replication. So although this is all correlation, this was our first strong evidence that late replication represents a stable epigenetic barrier to reprogramming. Okay, so when we get a large cohort, in this case it's mostly different cell types derived from human embryonic stem cells, but we also have some other cell lines and types peppered in here. what we can do is divide the genome into a large number of segments and then cluster them by replication timing. And so what you can see here, first of all, is that about a quarter of the genome is early replicating in all cell types, and about another quarter is late replicating in all cell types. And it's about 50% of the genome that shows some significant change in one or more cell types. Secondly, you can see that they cluster by their cell type, which is indicated here at the bottom. And lastly, we could identify what we call replication timing signatures, or RT signatures, of different cell types. And what these are groups of segments that replicate at times that are unique to that cell type. Now, some of you may be interested that another thing that we can do with this data is to create replication timing networks. The most interesting thing that came out of this work was that when we correlate changes in replication timing to changes in gene expression as embryonic stem cells differentiate to liver, pancreas, cardiomyocytes, neural precursors, we found that the genes that correlate best with these switches in replication timing are transcription factors. And in fact, they're transcription factors that are central to replication timing networks, or transcriptional regulatory networks. So we were then able to show that those transcription factors that are co-regulated with replication timing changes throughout the genome are actually co-binding at multiple sites within the domains that are switching replication timing. So we identified new interactions and provided evidence that it's transcriptional regulatory network changes that are driving these changes in replication timing. Now, I should mention that Anand Koren, who's now been at Cornell for quite a few years, is taking a different approach to identify what he calls replication timing QTLs, or RT-QTLs. And so what he does is, when he gets a cohort of individuals that are deeply sequenced and phased, what he can do is create replication timing profiles by just looking at the read depth, because early replicating sequences are at higher density than late replicating sequences in a given exponentially growing population. They have to be, of course, proliferating cells. And when he does this, he can find regions that are different between different individuals or different chromosomes. And he can identify variants that are linked to those replication timing changes. So out of 161 human B cell lines, this is now the 1,000 genomes cohort, he identified 16 loci with clusters of variants. Now, of course, there are many variants for each of these RT-QTLs, but it's an incredibly robust, actually, method, considering how low the dynamic range is when you have only maybe 10% of the cells proliferating. Okay, so what about disease? Part of the title of this meeting. So there's been many anecdotal correlations between replication timing and different genetically inherited diseases and disease-related phenomena like chromosome instability, also cancer and inflammation. And in one paper, it was found that disease genes in general seem to cluster in those timing transition regions between domains. And probably most of you know from cancer genomics that mutation rates and mutation signatures correlate with replication timing. For structural variation, we don't understand the mechanisms that well, but for single nucleotide polymorphisms, we now know that the mismatch repair proteins that run along behind the fork and clean things up are downregulated in the middle of S phase, so that early replicating sequences are well maintained, but late replicating sequences are mutated at a much higher frequency, and this is used to distinguish drivers and passengers in cancer genomics. And then recently, in an archive manuscript, Rishad Pavri has shown that in B cells, where the A protein mediates variation in immunoglobulins, it also can cause translocations between enhancers in other parts of the genome. And he's shown that if he disrupts replication timing, that actually reduces the frequency of these oncogenic translocations. So the hypothesis there would be that that genes that that replicate at the same time tend to be interacting, tend to be in the same location in the nucleus, so that they have a higher rate of translocation. And when you disrupt that replication timing, you disrupt the translocation frequencies. Okay, so I'm going to show you a couple of stories from my lab on disease. This first one is kind of a pet project. I also performed a sabbatical on this topic, and so this is a cancer study, and the cancer that I decided to focus on is B-cell acute lymphocytic leukemia, pediatric B-cell acute lymphocytic leukemia, which I'll call BCP-ALL. And the reason why I chose this disease, besides that Brian Drucker convinced me in a conversation early on to do so and give me some samples, is because it's a relatively, it's a very well understood cancer, but it has relatively very few mutations, so that results should be simple to interpret. And there are, of course, a lot of tools known, but it turned out to be kind of a grantsmanship mistake, because I continuously got the criticism that I was studying a well understood cancer, which really made no sense to me. But anyway, what I want to show you is here again, we're going to do this hierarchical clustering, dividing the genome into many 50 kb segments. And the first thing you should notice is that since all of these patient samples, this is a group of BCP-ALL patient samples with different genetic subtypes, etc. Some of them are what are called NOS, which means that they have no defined genetic subtype. Okay. But what you should notice right away is that there's only about 5% of the genome that varies, because these are so closely related. But nonetheless, we can take this 5% and then identify replication timing signatures that are specific for the different subtypes. And these subtypes do cluster by their genetic subtype. And then we can take these NOS patients, which are basically everything else. They have to be some kind of heterogeneous mixture of different types of BCP-ALL that we haven't defined yet. But nonetheless, we could cluster them separately and find that 2 of these patients shared a replication timing signature different from all the others. And these 2 happen to be relapse samples. And another criticism I got is, of course, that these children relapse at only a 10% frequency, so that I was studying a cancer that's virtually curable. Of course, then I would counter with the argument that if you were the parents of those children, I don't think you would agree with that argument. But nonetheless, of course, the scientific argument is it's a well understood cancer, and that's where we should start with a complex biological, novel biological phenomenon. Okay, so another thing we could do is take the patients that have the same genetic, patient samples that have the same genetic subtype, and cluster those. And as you can see over here on the right, we found a signature in common between 2 patients, both of which relapsed in the central nervous system, and not in common with remission patients. So these are very small numbers, of course, but I only was able to get very small pilot grants. I was never able to get a large grant. And so that's something I still need to work on. Oops, there we go. So now a question that comes up is, we see not only these replication timing changes that are in common between different subtypes of BCP-ALL, but we also see patient-specific alterations. And so a question that arises is, are these really stable characteristics of the cancer clone, or are they some kind of epigenetic stochasticity? For example, as Andy Feinberg has proposed a model of epigenetic instability or epigenetic stochasticity for cancer. We've done 2 things to address this. One is to take the patient samples and passage them through serially through 3 different patient-derived xenografts, and then query the replication timing profiles at each passage. And what you can see here is what the patient-specific replication timing patterns are conserved between the patient's original sample and the cells that are coming out of the PDX. So that says that these are extremely stable events. And we think they're epigenetic because of the very low mutational load in these cancers, but we, of course, don't have proof that they don't have some genetic origin. Another thing that we could do is, okay, so another criticism that I was taking during this project was that we didn't have a proper comparator, proper wild-type comparator. Now, we all know that that's very difficult in cancer biology, but with BCP-ALL, we actually have a good idea where the cancer derives from. It derives from the pre and pro-B stages of B-cell differentiation. But I was also being told by everybody that it's impossible to get these cells. They're very rare, and they don't proliferate. They're all quiescent. So you got to get them, but you can't get them. So I happened to be talking with Connie Eves on a seminar visit, and she said she thinks we can do this. So I took my sabbatical in her lab, and what I did was I took cord blood from normal individuals and bloomed them in xenografted mice, and then timed that so that I could collect the cells that are proliferating, that are at stages where the maximum number of them are proliferating, derived from these different stages of B-cell development. And in doing so, I could get replication timing profiles from these normal developing proliferating B cells and produce RT signatures. And down here is the conclusion of that, which is that the RT signatures in BCP-ALL are enriched for RT signatures that are in pre and pro, but not primitive hematopoietic cells and primitive lymphoid cells and not mature B cells. So from those two, we believe that these are very stable and characteristic epigenetic abnormalities in replication timing that are specific to each of these cancer clones. Okay, so the second story I'm going to tell you about derived from a collaboration we had studying replication timing in aging and senescence, and we didn't really find much in the way of replication timing changes in individuals' different ages or senescing cells. But in the process of this, Juan Carlos, whose name you see a lot, he's a very talented former postdoc who's now a professor at the University of Minnesota, and he was very active in my lab in the 2000 teens. So what he noticed in his reading was that there was a conundrum in the field of premature aging diseases or progeroid diseases, which is that we all know about progeria, Hutchinson-Gilford, but there's actually 11 different syndromes, and they all have very similar clinical traits, but they fall into these two general genetic categories, either the integrity of the nuclear envelope, most famous, the nuclear lamina, or DNA repair pathways. And there had been no common cellular trait found in common between these different classes of disease. So Juan Carlos identified RT signatures and found he could find common ones between Hutchinson-Gilford and Rothman-Thompson syndrome. And then he did something really clever, which is to reprogram these skin cells from these patients to iPSs, which erases all the differences between them. They all become normal-looking iPS cells. And then he redifferentiated them to fibroblasts, and he found that there's a gene TP63 that falls within the first replication timing abnormality to appear. And earlier replication in progeroid cells, either Rothman-Thompson or Hutchinson-Gilford, had altered promoter use, which increased the abundance of one of the TP63 isoforms. So just to summarize this, what we found is that in normal cells, this TP63 gene is late replicating and very lowly expressed and covered with H3 K27 methylation. But in progeroid fibroblasts, early in development, a part of this gene switches to early replication, and then this upstream promoter becomes more active and produces an overabundance of an isoform that has an extra exon and an extra N-terminus on its protein. Okay, so all of these sort of genomic studies, of course, are correlations. They don't tell us anything about the causality of replication timing to disease. So does replication timing actually influence chromatin? Is there a causal relationship there? And as I told you, it hasn't really been possible to test the hypothesis that replication timing can regulate epigenetic states because we haven't had rigorous ways to manipulate the program. And in fact, we looked at lots and lots of different types of chromatin regulators, and the result was either no effect on replication timing or very localized effects on replication timing. So what I'm going to show you is that has now changed, and we can now begin to address this. So in this slide, what I've summarized on the left is a lot of work that I didn't have time to tell you about, but basically this is our thinking about how replication timing is regulated in our thinking in 2021 anyhow. So this is partly speculative, but we a couple of years ago identified for the first time cis elements that regulate replication timing and regulate early replication, or causally direct sequences to domains to replicate early. And we call these early replication control elements, or ERCEs. And these ERCEs cluster in three-dimensional space, and they're coded with acetylated histones. So we think they form a three-dimensional hub of high levels of acetylated histones. This would then attract bromodomain proteins such as BRD2 and 4, which we know interact very strongly with a devoted replication initiation protein called treslin. So we would create this microenvironment in the nucleus that is poised to replicate early as soon as S phase begins. So what I'm going to show you today is that there's a protein called RIF1, which is coats the late replicating compartment of the nucleus. And that when we knock out this protein, we see first a complete or near complete, depending on the cell line, disruption of replication timing. And that's followed by delocalization of histone marks, and then disruption of transcriptional regulation and disruption of the 3D or architecture of the chromatin. And since this is published, I'm only going to show you, this is work that's done by former graduate student Peo Zhao and former postdoc Kyle Klein. Peo is now with Evan Eichler in Seattle, and Kyle is with the Allen Institute of Cell Science in Seattle. And as you can tell, they are a couple both in life and in science, and they are a couple to watch because they are really outstanding scientists. Okay, so I'm just going to show you two data slides. In this first one, what you're looking at the top is what we call high resolution RepliSeq, where we, in this case, took 16 fractions of S phase and isolated the nascent DNA in each of those temporal fractions, and then displayed that replicated DNA as a heat map. And you can see that the profiles look similar to the log2 ratio, but they're much, much sharper. There's a lot more detail here. But the point for this slide is that when we knock out RIF1, we get a complete loss of this program. And even this shadow of some sort of determinism in the background is not resembling at all the normal replication timing program. And so then we did CHIP. We did a lot of this work with Victor Corses. Victor Corses' lab did the CHIP and the Hi-C data, and we had really a wonderful collaboration with him. He's a very deep thinker. And so what I'm showing you here, we did a bunch of histone marks. I'm showing you here is just H2K27 acetylation. So you see that in wild-type cells. You can pick out peaks. We also showed these peaks generally interact with each other, and you can see that they align with the early replicating peaks in the RepliSeq. When we knock out RIF1, we get a delocalization of that signal, and we showed quantitatively it's a delocalization, not just a downregulation of acetylation, but actual delocalization of where these histone marks are. Here's a quick summary. So replication timing is disrupted in the first cell cycle. We then get misincorporation of histone marks, and those get worse and worse with each subsequent round, with each subsequent cell cycle. And then we see alterations in compartments, that is chromatin architecture, and gradual stochastic and cell type specific, we looked in different cell lines, effects on transcription that accumulate after several cell cycles. And as I'm going to come back to this, that cells actually maintain their identity and self-renewal. Last data slide. Just to give you one other piece of data from this paper to show you how we did this experiment. We moved to HCT116 because we could synchronize it, and we have a lot of degrons that have been developed and characterized in this cell line. So we took a HCT116 RIF1 aid degron, synchronized these cells in S phase, then either degraded RIF1 or did not, either held the cells in G1 phase or allowed them to go into early, mid, or late S phase. And what you can see over here, looking at two different chromatin marks, is that if we hold the cells in G1 phase, do not allow them to synthesize DNA, do not allow them to enter S phase, we don't see any changes in the histone PTMs, post-translational modifications. But as they enter S phase and go into S phase, we begin to see dramatic changes in basically depletion of peaks, or in the case of K9ME3, we actually see some peaks. Some peaks will become even stronger. And if we then go multiple cell cycles, we can see with each cell cycle we see more and more pronounced aberrations. And as I said, very few changes in chromatin architecture occur in this first cell cycle, and very few changes in transcription, if I didn't already say that. And these abnormalities accumulate with each cell cycle. Okay, so I'm going to leave you with a question here that we're very interested in pursuing, which is, why do we have an epigenome? These, as I said over here on the right here, if we disrupt replication timing, we disrupt the epigenome, but we don't affect self-renewal or cell identity. So we see that the core transcriptional regulatory networks are unaffected, and there are only minor stochastic transcriptome effects, especially in the beginning. I mean, these accumulate after a while, but the cells seem fine. But we also know that RIF1 null mice die at random points after gastrulation. So there is some essential role for RIF1, whether it's replication timing or not, we don't know yet. But our hypothesis then is that the epigenome is largely dispensable for cells to self-renew, but is essential for lineage commitment. So it's essential for cells to flip their epigenetic state and go from one stable epigenetic state to another stable epigenetic state. That's a hypothesis, and we can now use the exquisite human embryonic stem cell differentiation systems that have been developed to test this hypothesis, which is something that we plan to do. Okay, with that, I'll wrap it up. These are the people that helped us with that, demonstrating that replication timing is indeed necessary to maintain the epigenome, and in particular, Victor Corses and his postdoc Xiaowei Liu helped us really a lot, and our funding. And with that, I'll end and take questions.